Hello everybody, my name is Daniel Chichinsky. I'm a student with the Cropland Weed Ecology Lab at Montana State University. This presentation is on Thistle Rust, a potential biocontrol agent for Canada thistle. Circeme marvens is also known as creeping thistle, Canada thistle, or California thistle. It is an aggressive perennial weed found throughout temperate climates of the world. Canada thistle reproduces both sexually and asexually. Flowers can produce up to 5,000 seeds per stem. However, the plant reproduces most successfully through a network of vegetative creeping rhizome. The weed can cause significant crop yield loss, especially in organic cropping systems. It can also lead to a reduced diversity in natural and urban areas. The organism that we are looking into as a potential biocontrol for Canada thistle is a fungal pathogen called Puccinia punctiformis. We also call it thistle rust. Thistle rust is from the Basidio mycota family of fungi. It's highly selective to Canada thistle, where it exists as an obligate parasite, meaning that it requires living host tissue in order to complete its life cycle. It exists in the thistle rhizome as well as in above ground vegetation of the plant. Thistle rust lives a 5-stage life cycle, where 4 out of the 5 stages subsist by drawing resources from Canada thistle. The other stage is a dormancy stage, where the spores exist as leaf litter on the soil surface until they come in contact with a new host. The infection court would be a spore type known as teleospores, which are applied to thistle rosettes during autumn climates. So this image shows the pathogen life cycle. We can see that it's a pretty complicated life cycle. Thistle rust has been known about since the 19th century, and it was actually one of the first organisms mentioned in literature for its potential as a biocontrol. However, the complicated nature of this pathogen has really slowed its progress as a biocontrol until recently. The life cycle is really the foundation for understanding how to use thistle rust as a biocontrol for Canada thistle. So I'm going to go through an explanation of each stage and familiarize folks with what to look for in their fields. Stage 0 is called the Basidio spore stage. We can think of this as the vegetative stage in the life cycle. This is where the pathogen exists in the rhizome as a mycelial structure. Signs and symptoms include malformed leaves and chlorosis. However, this stage is thought of as asymptomatic because the plant rhizome might be infected with basidiospores while there are actually no symptoms that are visible above ground. This is the stage where thistle rust is drawing its resources from the host until it has enough energy to emerge with the shoot and begin the reproductive phase. Stage 1 is called the Spermata Stage. This would be the reproductive stage in the life cycle. Signs and symptoms include orange spores on the lower leaf surface of the plant. It emits a sweet aroma that might be linked to insect attractants. You also see malformed leaves as well as stem bolting. The season for this spore stage would be early spring to summer. Stage 2 is the aciospore stage. This stage has the potential to cause localized infection in new hosts, but it cannot cause systemic infection where it grows into the rhizome. Signs and symptoms would include friable spores, meaning that these spores are easily removed from the leaves and stems. They have dark red pustules on the leaves and stems. We see leaf chlorosis, necrosis, as well as bolting. The season for aciospores is this summer, depending on your climate. Then stage 3 is the Eurodynial spore stage. This stage can also cause localized infection in new hosts, but it cannot cause systemic infection. Signs and symptoms, once again, are friable spores, dark red pustules on the leaves and stems, malformed leaves, leaf chlorosis, and necrosis. The season for the Eurodynial spores would be late summer to early fall. And then the last stage is the teleospore stage. This is the dormancy stage in the life cycle where the spores have the potential to exist at soil level on dead thistle leaves and stems. Teleospores are responsible for systemic transmission of thistle rust into new hosts. Signs and symptoms include small black freckles on the leaves and stems, as well as premature leaf necrosis and abscission of leaves. The season for teleospores is late summer through the fall. And once again, this is the inoculation source that we use to re-inoculate new thistle in the field. So we know that the most effective way to suppress Candida thistle is by integrating management practices. Standalone weed controls don't provide long-term thistle suppression that we are looking for. Thistle rust is a naturalized pathogen which is co-evolved and highly selective to Candida thistle. However, it is not a silver bullet solution to the Candida thistle problem. Our goal is to explore the potential to integrate thistle rust as a Candida thistle biocontrol within organic agroecosystems. The studies that we are developing are focused on thistle rust's response to agronomic practices. In particular, we are looking into cropping rotations and tillage. For cropping rotations, we need to know is can we design cropping rotations that support the spread and establishment of thistle rust? And does thistle rust cause Candida thistle patches to decline at higher rates when grown with competitive crops? And for tillage, we need to know, does tillage, both conventional and reduced tillage, help? Does it hurt, or does it have no impact on the spread and establishment of thistle rust? So we've set ourselves a 3-year timeline. In 2020, we began by inoculating and surveying. This is where we established our experiments both in the greenhouse and in the field. We developed our experimental methodology, we tested inoculation methods, and we surveyed existing Candida thistle growth and infection rates. In 2021, we will continue to evaluate competition in tillage. We have seeded competitive annual crops both in the greenhouse and in the field. We've also begun splitting thistle patches into reduced tillage and standard tillage terminations. And we will continue to collect data on thistle growth and infection spread. And hopefully by 2022, we will be able to see thistle decline, which is caused by thistle rust and by these agronomic practices. And to do this, we will randomly place sample frames within our thistle patches. We will collect and analyze both crop and thistle density and biomass, and we will continue to evaluate the spread and establishment of infection within these patches. Fields at the Fort Ellis Research Farm. We set up experiments in 4 different fields. We mapped discrete thistle patches within each field, and then we counted the amount of symptomatically infected thistle within each patch. We then color coordinated the patches based on the amount of infected thistle that we saw. We then randomly assigned tillage treatments to each field. This was either reduced till, which is a flail mow, or conventional till, which was disc cultivation. In 2020, these fields were all seeded with forage barley. And in 2021, the fields will be winter pea and winter triticale. We are going to return in 2021 to reevaluate and hopefully see whether or not reduced tillage or conventional tillage practices have had any impact on the spread and establishment of thistle rust. The image on the left shows our flail mowing treatment at Fort Ellis, and the image on the right is our disc cultivation treatment at Fort Ellis. Okay, this image shows the setup of our field experiment at the Post Agronomy Farm in Bozeman. We have 2 fields. In each field, we planted thistle rhizome. There's a total of 20 control thistle and 20 inoculated thistle. The plans are to re-inoculate every year by applying a teleospore powder to thistle rosettes during fall climates. We will evaluate the effects of competition between inoculated and control thistle. In 2020, these fields were in fallow, and in 2021, we will seed forage barley as our competitive crop. We will also evaluate harvest and tillage between the inoculated and the control thistle. Our plan for 2021 is to swath and bale the forage barley within each field, and then we will follow this up with a mowing in order to minimize the regrowth of thistle. Data and analysis will include a collection of biomass and density of both thistle and crop, and we will also continue to monitor inoculations to determine whether or not our field inoculation methods are successful. This graphic shows the difference in thistle growth at our post-farm fallow fields in 2020. We had 20% symptomatic infected thistle only 3 months after inoculation. The inoculated treatment had less mass per individual compared to the control treatment. The red line shows the average mass per individual for all thistle in the study. We are pretty happy with these preliminary results. It shows that inoculations do actually impact thistle growth in fallow fields. Okay, we also designed a competition experiment in the MSU greenhouse. Here we planted thistle rhizome into 10-inch planters, and we simulated perennial rotations between an inoculated and a control treatment. Perennial thistle were grown to maturity, harvested, and allowed to regrow for multiple growth cycles. We then simulated cropping competition by mixing crops with thistle in the same planter. We made growth comparisons between monoculture thistle, monoculture crop, and mixed planters with both thistle and crop grown together. This trial had a total of 3 rotations. We had a simulated fallow, we had a spring wheat competition, and a spring pea competition. We then collected data and performed analysis on biomass and density of the thistle and crop, and we looked for any signs and symptoms of infection. And this image shows our study in the greenhouse. We can see thistle and spring wheat growing together in these 10-inch planters. So these are our preliminary results from the competition experiment in the greenhouse. The left-hand column shows our 3 cycles. Within each cycle, we had our control treatment and we had our inoculated treatment. Growth cycle 1 was our simulated fallow, where thistle was just grown in monoculture. So we don't have any results for competition because we didn't have a competition crop in those planters. In both our control and inoculation in cycle 1, we had 0% symptomatic infection. In cycle 2, we planted a competitive crop of hard red spring wheat. Our control showed 53% thistle biomass loss when grown in polyculture. There was 0% symptomatic infection. Our inoculated treatment showed 62% thistle biomass loss when grown in polyculture, and there was 20% symptomatic infection. Then for growth cycle 3, we planted our Vika forage peas as our competitive crop. We had 61% thistle biomass loss in polyculture in the control treatment, and again 0% symptomatic infection. Our inoculation group had 67% thistle biomass loss when grown in polyculture and 10% symptomatic infection. What this means is that Canada thistle produces much less biomass when it's grown with competition versus grown in monoculture. Additionally, these results show that thistle rust actually causes even higher losses in thistle biomass within competitive cropping systems. So integration of thistle rust in agronomic practices will require crop selection and tillage, which facilitates establishment and spread of symptomatically infected Canada thistle. Again, thistle rust is not a silver bullet solution to the Canada thistle problem. However, we believe that it can be impactful when it is intentionally added to a farmer's IPM plan. Continuation of this work will clarify the ecological interactions between thistle rust and agronomic practices, and hopefully help determine whether or not this pathogen is actually a viable biocontrol agent for Canada thistle. So I'd like to thank everybody who helped make this possible. We have an incredible network of people who have been a great help to us. Unfortunately, I will not be able to make it to the December 5th MOA meeting. So if anybody has any questions or comments, then please scan out this QR code or select the link, and it will take you to a Google form where you can fill in your comments. We have also added an optional survey. Please fill out the survey if you have the time, and we would really appreciate it. Thank you all for listening, and have a great winter.